It hangs in the dark. A silent, ghostly form drifting miles below the surface. This is not a known entity, not a familiar shape from any textbook. It is a new transmission from the abyss, a biological puzzle captured by the Monterey Bay Aquarium Research Institute, or MBARI. What are we looking at? This footage, captured by a deep-diving, remotely operated vehicle, shows a creature that has perplexed the Institute's scientists. It appears to be a new species of jelly, one that does not yet have a name, an animal awaiting its place in our scientific registry. Its body is a cascade of delicate, thread-like arms, far more complex than a simple jellyfish. But it's what those arms are doing that has captured our attention. They seem to glow with a purpose, a cold and deliberate light. This is not a passive drifter. This is an active and likely highly specialized predator. We are witnessing the very first encounter with a life form that has mastered its dark environment in a way we may have never seen before. This is the frontier of discovery, happening in real time. Now let's dive into the data and uncover the reality hidden in the deep. This first glimpse was not a chance encounter in shallow water. It occurred at a depth of 990 meters, deep within the Monterey Canyon. The discovery was made by the team aboard the RV Western Flyer, deploying their key instrument, the remotely operated vehicle Doc Ricketts. The Doc Ricketts is not just a camera. It is our remote set of eyes, hands, and scientific instruments, allowing us to be present in a world we cannot physically survive in. The ROV was conducting a transect, a systematic survey of the Midwater or bathyal zone. This is a realm of perpetual twilight fading into total darkness, immense pressure, and cold, stable water. Life here is sparse, and encounters are rare. Then it simply appeared. A form drifted into the vehicle's high-definition lights. Immediately, the trained pilots and scientists in the control room knew this was something unusual. It didn't match the typical profiles of the ctenophores or siphonophores that dominate this depth. The creature was estimated to be about 10 centimeters or 4 inches across. It moved with a slow, deliberate pulsing. But the most striking feature was its intricate arrangement of arms. They were not the simple trailing tentacles of a common jelly. Instead, they were held in a complex, almost rigid posture, radiating outwards. This was not a known morphology. When we, as researchers, encounter an animal like this, the first step is a frantic mental search through a visual library. We try to place it in a family, a genus. Aurelia. No. Atola. Not even close. This creature's body plan was so distinct that it resisted easy categorization. The team on the flyer recognized this. They skillfully maneuvered the ROV, zooming in to capture every possible detail. They documented its movement, its shape, and crucially, the faint pulsing light emanating from those bizarre arms. This is the critical moment of discovery, not a eureka shout, but a quiet, intense focus, gathering the data that will form the foundation of a new chapter in biology. The ROV's collection system carefully took a sample, securing the specimen for retrieval. Without this physical specimen, it would remain a ghost, an anomaly on a screen. With it, the real work of identification, of understanding, could begin. Once the specimen was secured and brought to the surface, the deep dive analysis began. The high-definition video was just the first piece of the puzzle. The true deconstruction happens in the lab, under microscopes and inside genetic sequencers. And the central focus of that analysis was the arms. They are not simple tentacles. In a typical jelly, tentacles are the primary weapons, loaded with stinging cells or cnidocytes to shock and capture prey. This creature's arms appeared far more delicate, almost like gossamer threads. Yet they were held in that specific, rigid, radiating posture. This rigidity suggests a high degree of muscular control, not the passive drifting we associate with jellies. And then, there is the light. When we analyze the phenomenon, we are talking about bioluminescence. This is a chemical reaction, a cold light produced by the animal itself or by symbiotic bacteria it hosts. The ROV footage captured a faint, rhythmic pulsing. This is not a defensive burglar alarm. That's a bright, sudden flash meant to startle a predator. This is not a luminous goo defense meant to confuse an attacker. This light was controlled, it was sustained, and it was integrated directly into the animal's hunting apparatus. The conclusion became inescapable. This is a lure. In the pitch black of the Bathyal zone, light is the single most powerful form of communication. It's also an enormous metabolic cost. An animal does not waste energy creating light unless it serves a purpose critical to survival. For this creature, that purpose appears to be predation. It is a bioluminescent hunter. We can now build a hypothesis of how this animal lives. It likely hangs motionless in the water column, a living minefield. It activates its photophores, the specialized light-producing organs in its arms. The faint, pulsing light is designed to do one thing: attract prey. What kind of prey? The light may mimic the bioluminescent signals of copepods or small krill. This, in turn, would attract the slightly larger animals that feed on them, perhaps small fish or other crustaceans. In the absolute dark, any pinprick of light suggests a potential meal. This jelly is creating a constellation of false promises. As the curious prey swims in to investigate the copepod, it bumps into one of the nearly invisible, filament-like arms. These arms are almost certainly loaded with a capturing mechanism, either the stinging cnidocytes of a jellyfish or the sticky colloblasts of a ctenophore. The prey is instantly trapped, and the jelly retrieves its meal. This strategy is not entirely new. We know the deep-sea anglerfish, which dangles a single bacteria-filled lure.